Yeah. Okay. Uh, 有人是下午好哈，
下午好，
你好。那因为我们今天当初也是说我们就是全程用英文，所以我开始我就 switch 到那个 English 的部分哈。OK，
问题对。
It's our great honor to invite the Fudong Liu Academia to have the talk today. And because everybody know right now is the COVID-19 outbreak, so we don't want to block any possibility for the master symposium. So today we invite Professor Fudong Liu give us a nice talk. And let me introduce the Fudong Liu a little bit. He is a chemist, but also is a medical doctor, and he focused on the glycoprotein research maybe 20 years and have a lot of the outstanding finding. And Professor Liu as the academia during after the 2012 in academic Seneca. And he also, as I know, you are the president of the immunology societies right now.
Right.
Yeah.
Oh, actually, I just finish my term.
Finish your term.
Right.
Okay.
So the new president is a Situ.
Okay. Because next year is the physiology society in charge for the biomedical conference.
Yes,
so
right.
I read some information as I know. Okay. So today's we have like a 1 hour's talk. Let the Professor Liu to give us about the glycan binding protein from the biology and therapeutic potential. So give the warm hands to welcome our Professor Liu.
Thank you. I want to thank慈济University College of Medicine for this very kind introduction and allow me to share with you my knowledge in glycobiology and share with you my research. So it's unfortunate because of the COVID-19 situation, I'm not able to visit in person. I hope when the pandemic is better controlled, I visit the campus again. So my topic is glycoscience. So 2012, less than 10 years ago, or almost 10 years ago, the National Research Council of National Academies, this is the United States, coordinated jointly by National Academy of Sciences, NAS, National Academy of Medicine, or maybe also with engineers. They jointly prepare a document entitled Transforming Glycoscience, a roadmap for the future. And they state that it means if we transform glycoscience according to the roadmap proposed, it can make contributions toward understanding and improving human health, as well as many other benefits, including, I remember that even a generation of new materials. And they emphasize that now, meaning that at that time, is the time for glycoscience to be embraced, these keywords embraced broadly by the research community. And they say that although these challenges are substantial and complex, and you might get the impression that indeed the area I'm going to talk about is a little bit complex. But the results of achieving these goals have the potential to impact science in exciting ways. Then just merely about 5 years later, meaning about 3-4 years ago, Nature, there's an article published in Nature, Sweet Success. And it's highlighted that biologists are diving into sugar molecule research thanks to new tools and techniques. Those new tools and techniques are glycomics tools and techniques. And by glycomics, this is just a very nice photo or diagram of what glycobiology is about. That's we have glycoproteins and include O-linked glycans and N-linked glycans. Also glycolipid and glycosaminoglycans, proteoglycans. Inside the cell, there's a very important glycosylation. That is O-linked N-acetylglucosamine. This is just monosaccharide. Hundreds or even a large number of intracellular proteins are glycosylated this way. Just like many are phosphorylated. In fact, the site for phosphorylation is this phosphorylation on serine and threonine, not on tyrosine. Serine, threonine phosphorylation and the O glycosylation interchangeable, meaning that the site that can be phosphorylated can also be glycosylated. They can go back and forth. But, I will not talk about this glycosylation today. And also like to mention that for the purpose of today's presentation, and also for you to, when you get encounter in this area, just think about this glycan as a pattern or as some kind of shape. Just like when you look at proteins, you don't necessarily think about amino acid sequence. You think about maybe one kind of shape, and there's a binding site that the bind ligands. Same thing. So I know that many of you might not be familiar with glycosylation, but these glycans, actually, when you think about it, don't think about complex structure. You think about this as a pattern, especially for today's presentation. So I mentioned that many of the tools and techniques are developed and making actually this sweet success. Glycomics is the systematic study of all glycan structure of given cell type or organism. However, just like when we talk about proteomics, we learn about we determine the structure of proteins. We do not necessarily know the function unless the protein is homologous to some known protein with a known function. Same thing for glycomics. So when you determine the glycan structure, you don't necessarily know the function. So there's an area called the functional glycomics. So the authors stated that to decipher the function, we need to identify proteins that recognize the glycans that can translate information encoded by a specific glycan structure. So the functional glycomics was named to define the concerted and regulated action of carbohydrate binding protein or glycan binding protein, which is topic of today's talk. I don't quite completely agree with this definition because I think glycans, they have other functions in addition to being recognized by glycan binding proteins. For example, they can keep the protein, the glycoprotein, in certain shape, certain the stability and certain conformation. However, I think this is a very convenient term to talk about how we move from glycomics to functional glycomics, and that is the study of glycan binding protein. Indeed, this is the very major branch of glycobiology. In fact, it's also one direction with less barrier in terms of getting into glycoscience by studying these glycan binding proteins. However, I mentioned today's at the interview in the morning that after today's talk, by no means I wanted everyone to jump into glycoscience. You have your professor have your research topic, and the student have their thesis or their learning, still learning science, and but these days it's the way research is being conducted. Usually, we go through high throughput screening or non-biased survey to get the overview of what's going on in the system you're interested or the disease you're interested in studying. Then, if through that exercise there are some glycan binding proteins that surfaced, then I would like you to embrace that opportunity and just say that, well, this is actually opportunity to get into glycoscience. In fact, I got into glycoscience totally by accident, but I won't go into that today. So these are major types of glycan binding proteins. Glycan binding protein, another term is old term is lectins. Lectin in Chinese is Ningji Su. So glycan binding proteins are lectins. They are, I think you are familiar with many different lectins, especially plant lectins. So I'm sure you're familiar with selectin. There are 3 selectins: L-selectin, E-selectin, and P-selectin. These are very famous glycan binding proteins. And these are C-type lectins. C stands for calcium. So these are the activity require calcium, the presence of calcium. Then there are hundreds or even maybe over 1,000 C-type lectin like receptor, C-type lectin. Put hepatic lectin here. I won't get into this very large family proteins. Then another interesting and important family is the Sig-like or I-type. So I will talk about this later. And galactans, which is the major topic of today's presentation, different from other lectins in that they are not transmembrane proteins, and they do not have so-called leader sequence or signal sequence. So they are not necessarily made to be secreted. So they are actually reside inside the cell, although you can find that outside the cell. This make the study of galactans a little bit challenging because they are, as you see, the glycoligand recognized by all these lectins reside outside the cell, on the cell surface and extracellular matrices. Although they are made inside the cell, right? But they are expressed outside. And galactans, glycan binding protein, but they reside mostly inside the cell. I will dissect that, what they do later. So I want to share with you challenges associated with investigation of glycan binding proteins. So I mentioned that from proteomics to protein function require our understanding of what the protein bind to or interact with. So take cytokines, for example. Each cytokine has its unique receptor. So many of cytokines you're familiar with, or we call interleukins, or even some mediators like hormone, they have their unique receptors. The glycan binding proteins are different. So when we go from glycomics, we know to functional glycomics, we go from we know the glycan structure to its function. There we definitely can connect to glycan binding proteins, but given glycan binding proteins may engage different glycoproteins on the cell surface. because different glycoproteins may express the same glycan ligands. So that is glycan binding protein bind glycans. The glycan can be carried by many different glycoproteins. So when you engage that glycan, maybe many glycoproteins are engaged. And also, when glycan binding protein get outside the cell, you actually might not be able to predict what glycoprotein you will bind to, because there are many possibilities for them. So I will talk about selectins, which you more familiar with, and then Siglex, and then spend about the second half of my talk on galactans. There, I talk about extracellular function first, and I'd like you to remember I have a question mark here, and then please, I hope I can explain clearly why this question mark. Actually, this has to do with the previous slide, that is a challenge associated with this field of research. Then I will spend most of the time on intracellular function. There, I mentioned that inside the glycoligands are outside the cell, but so inside the cell, actually galactans can function in glycan independent fashion. And I will share with you our study on galactan 3 and disease, and galactan 7 as relationship to psoriasis. But then share with you, I believe, very exciting development in the field of galactan, in fact, a field of glycobiology, that is galactans, may be unique in this aspect. That is, they have glycan-dependent function inside the cell, inside the cell, intracellularly. So selectin, as I mentioned, that this is a C-type lectin. C stands for calcium. It's calcium-dependent. And the L-selectin, E-selectin, and P-selectin. This is almost among the first family of glycan-binding protein that were discovered, and they actually enhance the appreciation among the scientific community of the importance of carbohydrate binding. And these are themselves glycoproteins, but these are lectins, but they're themselves glycoprotein. Lectins are not necessary glycoproteins, right? But these three selectins happen to be glycoprotein as well, and they bind to carbohydrate presented on glycoconjugates, and they are very well known for being cell adhesion molecules. They mediated cell adhesion cascade, and during inflammation, selectin serve as these biomolecules that are responsible for trafficking of leukocytes from their origin to the site of inflammation. So this is already a very classical diagram, and this is pretty much like a textbook, something you will read in standard textbook. So P-selectin and E-selectin, P actually stand for platelet, but they also expressed by endothelial cells. E stand for endothelial cells. So during inflammation, if this site is going to have inflammation, P-selectin and E-selectin are induced. Then they bind Sialyl-X modified glycoprotein. I'll come back to this in the next slide. I got 2 slides next reversed. So by recognizing glycan, this leukocyte got attracted to the site. And this is a low affinity interaction. So this leukocyte will roll on this endothelial cell, on the endothelium surface. But then as the inflammation signal continue to evolve, there's other ligands, other biomolecules expressed or induced. For example, ICAM1, and these are ligand for integrin. So the integrin on leukocytes will bind to this ligand, and this is firm adhesion. So now the cell was kind of stopped there, and then later on, traverse through the gap created between endothelial cells, and they get into the site of inflammation. So after discovery of selectin was really big news in scientific research, and also started to get glycobiology recognized by the scientific community. Then the next important progress was the discovery of the glycoligand recognized by selectins, and that is Silu-X, this tetrasaccharide. Then the scientists move on to try to find a receptor for selectin, and for the first one that was discovered was PSGO1, so P-selectin glycoprotein ligand 1. And it turned out these are recognized by all three selectins. And later on, there are more and more glycoprotein ligands discovered that bind to selectins. So actually, this is the point I made earlier in terms of there's no one-to-one relationship between glycan binding proteins and its glycoprotein counterpart, because unlike protein cytokine and cytokine receptor interaction, right? So actually, I believe in the selectin field, people do not talk much about what they bind to. They mainly study their function. This issue will become more obvious when I talk about galectin. Nevertheless, the field of selectin continue to evolve, and almost 20 years later, I think this is the first drug targeting selectin. It's a P-selectin. So these are two professors from University of Oklahoma funded this company, and they founded it because they study P-selectin, and they have antibody against P-selectin, monoclonal antibodies, and I believe this will be useful to maybe has post-therapeutic potential. That was 2002. Almost 10 years passed, and they actually started to work on something they never expected they would get into, that is sickle cell disease. And they came up with the notion that P-selectin may be involved in mediating adhesion of sickle erythrocyte to the blood vessel surface. So indeed, clinical trial using this monoclonal antibody, they show that blockade of P-selectin by the monoclonal antibodies they have reduced sickle cell related pain crisis, or this called the vaso-occlusive crisis. This really very patients very much suffer from this disease. They almost have to go to emergency room each time there's a pain crisis. And the company was acquired by Novartis in 2016. And then this drug was approved by US FDA in November 2019. So 17 years after the company was funded. And this is actually a success story. I know the founders well, especially one of them, Richard Cumming. He later on moved to Emory and moved on to Harvard. He's now a program director at Harvard, a very successful glycoscientist. Siglec. So Siglec is sialic acid binding immunoglobulin-like lectins. So SI is sialic acid. Sialic acid is a monosaccharide, has 9 carbon instead of 6 of the other monosaccharide. So IG is immunoglobulin, and LAC is lectin. So Siglec. It contains one domain that binds to sialic acid, and the other immunoglobulin-like domain can be 2, 3, or many. I will come back to that, talk about that a little bit later. So in the cytoplasm, there's immunoreceptor tyrosine-based inhibitory motifs, or ITIM. You know, there are ITAM and ITIM. ITAM is activating motif, and ITIM is the inhibitory motif in the cytoplasmic cells. So this implies that these proteins or glycoproteins play a suppressive role. They downregulate intracellular signaling pathways. There are 15, but in fact, the 16 is Siglec now. There are roughly two groups. One is conserved, meaning that if you want to study the mouse Siglec, you can find the mouse Siglec corresponding exactly to the human Siglec. They are CD33 related Siglecs. They are not conserved, and actually they evolve very quickly to the point that when you study one Siglec from in mice, you might not know exactly which Siglec in human it corresponds to. This caused some challenge to this field, because when you make mice without giving Siglec, which is a very common approach these days, you do not know the phenotype you observed tells you about what the function of the given human Siglec. Also note that this red circle ITIM motif. So most of Siglecs contain this ITIM motif. Siglecs are studied mostly in hematopoietic cells, leukocytes, and each leukocyte express one, two, or many Siglecs. And as I mentioned, that they are suppressive. So, for example, if this is B cells, when the activatory receptors, for example, this could be base cell receptor, immunoglobulin, when they are engaged, they mediate activation signal, or they transmit. The activation signal get transmitted. But if a Siglec, in this case a Siglec3 or CD33 is also engaged, then somehow they will recruit phosphatase, and phosphatase is known to cleave phosphate in this in the signaling pathway, molecules in the signaling pathway, and leading to suppression or inhibiting or dampen this cell activation process. Just share with you one recent, very interesting, exciting development in the field of Siglecs. There are many examples. I'm sorry, I have time to talk about just one. So NK cell are known to express Siglec7, and tumor cells known to express salicylic acid carrying glycoproteins. And in this case, these authors show that some glycoproteins on tumors can bind to Siglec7. Then, because Siglec7, like many other Siglecs, are immunosuppressive molecule, and so the NK cell function of NK cell is a natural killer cell. They can kill tumor cells. So interaction of salicylic acid ligands on tumor cells, they shouldn't be tumors, should be tumor cells on Siglec NK cells may dampen NK cell activation in a tumor context. So I think you're familiar with PD1 or CTL4. That's the Nobel Prize 2 years ago. PD1, both are immune checkpoint, and immune checkpoint blockade now is the mainstream of therapy, cancer therapy. But we also know that they only a subset of cancer, of given cancer, can be treated or can be cured by this approach. There are other immune checkpoint molecule, and the blockade of those can maybe used together or maybe separately for treating cancer. This is a new direction in cancer therapy or cancer immunotherapy. And this Siglec7 blockade might be one way. And there are other Siglec being discovered to have this immune checkpoint-like function. And I think I'm going to skip this one, just say that another therapeutic potential, this was published 2013, and I'm not sure this has become, whether the investigators are continuing to pursue this possibility or not. They generate this liposome containing actually antigens recognized by IgM on the B cell surface, and they also have molecules that can bind to CD22, which is Siglec-2, and the Siglec-10 or G, and then cause the separation of B cell function and antibody production. So remainder time I will talk about galactans, 半乳糖凝集素. So galactans are beta galactoside binding animal lactans. So gal is a galactan, galactose, and lactan. So galactose binding animal lactans with conserved CRD, carbohydrate recognition domain. They evolutionarily highly conserved, a little bit different from Siglec. Siglec evolved quickly to the point that so mouse Siglec can look very different from human Siglec, although they still share homology. They are found in nematodes. So very commonly used model organisms is C. elegans, is nematode. They express quite a few of galactan family members. And all mammals express various galactans. Galactans can be one CRD, they exist as a homodimer, or two CRD in tandem, and this is separated by a short peptide linker. And galactans, we actually were the first to, my lab is the first to clone and sequence galactan 3. Back then, this is the galactan family was not known, did not exist when we first cloned and sequenced the protein later on known to be galactan 3. I'll talk about the 7 mainly and galactan 3 and maybe 8, and very quickly on 9 and 12. So galactans, in addition to binding galactose, they also share some signature sequence or consensus sequence highlighted here, and these are invariably present in among all galactans. However, the sequence identity among different galactans are actually not that high. So, for example, between 1 and 3, 1 and this part of 3 maybe just about 40% or less. And but between human galactan 3 and the mouse galactan 3, human galactan 1 and mouse galactan 1, there's maybe 80%, 90% identity. So it's highly conserved. For that reason, when there are so many members, one commonly asked question is whether there's redundancy. But I don't believe there are redundancy. Maybe between 1 and 2, but the others are very different among each other. So some galactans have wide tissue distribution. For example, galactan 3, I will talk about expressing many different tissues and cells. Others, like galactan 7, I will talk about, and especially galactan 12, they have higher tissue specificity. I mentioned that the galactans are not transmembrane protein. They don't have transmembrane domain. They do not contain, or they are not synthesized with classical signal sequence or leader sequence. So they are not made through the Golgi pathway and exocytosed, get outside the cell. But consistent with that, they are found mainly in the cytosol. They can go to nucleus, but also in the extracellular space. We do not know exactly how they are secreted because they do not have classical signal sequence. Like many other lactans, 凝集素. So galactans are bivalent or multivalent with regard to their carbohydrate binding activity. So they can cause aggregation of cell glycoprotein. They can cause aggregation of cells, although we are not sure aggregation of cells of the function of this protein inside the body. For example, this galactan can cause hemagglutination. So this is a very convenient way to test the activity of galactans if you purify it from as a recombinant protein or from cell line. But this does not mean this is the hemagglutination is the activity of one of the function of galactans. So you might find that this field is somewhat complicated, but a good thing is they are getting more and more attention because they are implicated in the pathogenesis of a variety of diseases. This is mainly in recent years because now many glatins have, scientists have developed mice that lack a given gelatin. So they are more and more knockout mice. And from there, by studying the phenotype, they have been implicated in the pathogenesis. So this is the something I wanted to share with you. I mentioned that the question mark following the extracellular function of galactans. So a variety of cellular responses can be induced in vitro by using galactans added exogenously to various cell types, because this is a multifunctional protein that bind glycan. So they can envision that they can cause aggregation of or cross-linking cell surface glycoprotein. And usually this means transmit signals, signal transduction, right? And so this is easily understandable. Then if you go one step further, and indeed the researchers do that, have done that. That is, you inject galactans into experimental animals. Then you find some response. However, and to mention that these may not represent the function of endogenous galactans. Issues, one is, first the easy one is galactans might not be secreted or secreted in sufficient amount. So they might require a larger amount to induce activation of cells or cause response of cells, but they may not be secreted in the first place, or they may not secrete in sufficient amount. Then specificity is the point I mentioned. So unlike cytokine has unique cytokine receptor, the one-to-one relationship, the galactans, they do not have one-to-one relationship with a given glycan. So from the in vitro study mentioned on the top, we really cannot predict what glycoproteins on cell surfaces or extracellular matrices galactan bind to. If it's secreted in sufficient amount, we cannot predict where these galactans will go to or which cells they will bind to. I hope this is clear. I know this might be a little bit complicated, and over the years I try to convey this message. I'm not sure I've been successful. Although I think it shouldn't be a too difficult concept. So for many years now, we actually used to just add galactan 3 to cells to see what happened, and conclude the function that way, and publish paper. In fact, I regret that we did that. But then subsequently for many years now, we want to ask the function of endogenous galactans. Endogenous 内源性 does not mean it's intracellular, but it's just the cells come with this galactan. We want to ask what the function of those galactans they come with the cell, rather than what happened when you add recombinant galactans or purified galactan to cells. Then we ask whether, because galactans are mainly intracellular protein, whether they have intracellular functions. So we actually have been lucky that we succeeded in identifying a number of function of endogenous galactans, and these are intracellular function. Although each one took quite a few years. So the first one, this is the first intracellular function of galactan, is the anti-apoptosis. Anti-apoptosis, this is in T cell. And in that work, we show also galactan-3 bind to very well known anti-apoptosis protein, BCL2. So we propose that galactan-3 exert anti-apoptosis function by binding to BCL2. And the next one, this is very interesting, and I'm always very pleased with how we came up with this, the findings that gelatin firstly promote phagocytosis. This is in macrophage. And then when phagosome forms, when phagocytes or macrophage engulf certain particles or cells, and then this substrate became part of phagosomes. Then gelatin-3 recruited to surround these phagosomes. Phagosome is on the cytosolic side. Then later on, we found in T cells, gelatin-3 also translocate to cytosolic side of immunological synapse, and this is involve T cell receptor, and through T cell receptor engagement, the signal can be transduced, and gelatin-3 can modulate that signal. And then we implicated ALEX that we found to be a gelatin-3 binding partner. Through binding to ALEX, gelatin-3 modulate T cell receptor mediated signal transduction. And then there are other findings later. We published, in fact, there are maybe 3 or 4 more. And so here, I should, okay, so something is blocking. Okay, yeah, maybe this helps. Interaction with the intracellular interaction with intracellular regulators. So these are intracellular function. And inside the cell, I mentioned earlier, although they are glycoprotein, glycolipid, they are in the ER and Golgi on their way being synthesized and then get outside the cells. But in the cytosol, there's actually, other than that old gluconeck I mentioned earlier, there's no glyco conjugates or glycoprotein, glycolipid in the cytosol. Okay. I should mention that the gelatins do not bind to O-linked N-acetylglucosamine, which I talked about earlier. And this is so this binding is independent of glycan binding. So for example, BCL2 and the ALEX, they are not glycoproteins, but gelatin-3 interact with them. However, the glycan carbohydrate binding site domain may be involved because this is a small molecule. I should have mentioned that each CRD is maybe about 120, 130 amino acid, and the carbohydrate binding site can be used for binding to protein, which is not too surprising. Okay. So then we have so many of this work I mentioned was we used the cells from gelatin-3 knockout mice we generated, and we also studied the phenotype of gelatin-3 knockout mice mostly in allergic skin and airway inflammation, atopic dermatitis, contact dermatitis, and asthma. And all these three, we found that gelatin-3 knockout mice have reduced or lower responses, and the pathways that involved the gelatin-3 knockout mice have a lower response. So gelatin-3 in this in vivo experiment, we show that gelatin-3 is critical for T cell mediated allergic skin and airway inflammatory responses. There are many other papers published by other investigators, and also we have published a few others as well. So in general, gelatin-3 is a positive regulator of a variety of inflammatory diseases. Although there are exceptions, they may be pro-inflammatory, maybe they are inhibitory, but largely, mostly, this is positive regulator. Very interesting that gelatin-3 now is well known to contribute to chronic heart failure. So this was first discovered by gelatin-3 being a marker, actually in the blood, that correlate with the prognosis in patients with chronic heart failure. So actually FDA approved the use of ELISA kit to measure the gelatin-3 level, and then allow the physician or cardiologist to predict the prognosis of the patient, and then that might guide the treatment. in fact, I just did a search for keywords heart failure and gelatin just a couple of days ago, and there are already over 600 papers. So this, I believe, in the cardiology field, this is pretty well recognized. And there are a couple, but however, I found that the mechanism is not known until recently. So these are two papers talk about gelatin-3 contribute to inflammation and fibrosis. These are both mouse models, and they use gelatin-3 knockout mice. However, I mentioned that the gelatin-3 is first found to be serum biomarkers. So many investigators would think that they are some kind of mediators. They could be functioning like cytokine and to lead to inflammation, fibrosis. However, that might not be the case, and that's what I mentioned about the extracellular function. And so these two papers actually did not address whether extracellular, but mainly talk about the phenotype of mice. So this is still work to be done, and this is important because if you want to find develop inhibitors and for treatment of heart failure, you need to know where gelatin-3 function. And similarly, in renal diseases, there are papers already published about gelatin-3 contribute to fibrosis. So I found nice review articles on gelatin-3 in autoimmune and non-autoimmune nephropathies. I need to mention that when you read a review like this, in fact, include the review we published earlier, some of the conclusions made might be related to the extracellular function that published earlier. So you need to kind of dissect what is the information contained in the review in terms of which one is result from the study of endogenous gelatin, which one is study of exogenously added gelatin. So from all this research, gelatin-3 may be targeted for treatment of inflammatory disease. That's related to the title of today's presentation, the therapeutic potential. But then I ask, should we target intracellular or extracellular glatins? Okay. So I think you probably guess what my answer is. Actually, there are several companies working on gelatin-3 inhibitors or developing therapeutic based on inhibiting gelatin-3. The most successful one is Galacto Biotech. This is the company established by a professor from Lund University. They are a chemist and biochemist. They started this company. Started with making inhibitors for gelatin-3. The TD139 here is now very well recognized, very well known. In fact, it became commercially available. It's a derivative of TDG, that is a thiiodiglycoside. And the company has succeeded in showing in phase 1, and I believe it is now in phase 2, that the TD139 was effective in treating very difficult disease to treat, that is idiopathic pulmonary fibrosis. So I talked with the company, and they are not quite sure how TD139 work in blocking gelatin-3 function. But we know, and they also know, that TD139 is cell permeable. So it can get inside the cell and block gelatin-3. If gelatin-3 is for the pulmonary fibrosis, gelatin-3 working inside the cell, then it can work as well. If the company started by immunologists, then they probably will be just like the other company on Selectin. If they probably would have started with anti-gelatin-3 monoclonal antibody, then I wonder whether it will work or not. But it's just my biased opinion. Okay, so quickly on gelatin-7 and psoriasis, mostly is our work. And psoriasis, I think you are familiar with, so I won't read this in the slide, but just say this is another inflammatory disease, and there's a proliferation of keratinocytes, epidermis, and also inflammatory infiltrate. And I hope you know Th1, Th2, Th17. This is mainly Th17. Also, I want to emphasize this is a metabolic syndrome, meaning that the patients usually obese and they have dyslipidemia. I will make come back to tell you why it's relevant. So these clinical slides, a patient can have over 80% or maybe full body covered by psoriatic lesion, and this is a very typical psoriasis plaque, psoriatic plaque. And you don't need to do biopsy to recognize this, especially dermatologist. And but if you do biopsy, you find this so-called psoriatic form hyperplasia. And then if you stain for leukocytes, there's a dense cell infiltrates, both in dermis and epidermis. So Th17, meaning that dendritic cells are known to drive T cell into Th1, Th2, Th17 direction polarization, and depends on the cytokine they produce. So if it's IL-23, they drive to the Th17 direction. And either Th1 or Th17 cytokines can then cause keratinocyte to be activated and proliferate, and then subsequently this will lead to generation of psoriatic plaques, and because there's cell proliferation and also increased inflammation, because keratinocyte can actually mediate the inflammatory response. And this is another way to look at psoriasis. This is a little bit out of date, but in general, we know that dendritic cells play an important role, and through the Th1, Th17, mainly Th17, T cells and cytokines, keratinocytes get proliferated, activated, and they can in turn attract and activate neutrophils, and all come back to they come back to activate dendritic cells. So this is the amplification process here. And so Glan7 is expressed in stratified epithelial cells like skin keratinocytes or esophagus. They're highly induced by p57. Galectin-3 is the anti-apoptotic. Galectin-7 we show that it's pro-apoptotic and is tumor suppressor. Another group showed that galectin-7 is bind to BCL2. We show galectin-3 bind to BCL2, but galectin-7 also. The right side is mainly just to impress you that try to impress you that this intracellular protein and also can be highly enriched in nuclei. So we work on psoriasis. The work is mainly done by two postdoc fellow, Chen Hongling and Luo Jiahui. The reason we got interested in galectin-7 is we conducted a transcriptomic analysis with Genentech, a company, biotech company, and the paper is already published. We look at atopic dermatitis, contact dermatitis, and psoriasis. For each one, we recruit 10 to 15 patients and the biopsy the non-lesional and lesional skin, and look at the gene expression profile. And for psoriasis, we found galectin-7 is downregulated in the lesional skin, downregulated. And this was confirmed by immunohistochemistry. This is lesional, non-lesional, and less lesional. You see, it's less intense staining for galectin-3. So our question was what happened to the how galectin-7 downregulation through downregulation cause the psoriasis development psoriatic lesion through keratinocyte. And we have this already published. So we found galectin-7 actually can regulate, modulate expression of actually only 4 microRNA. So microRNA 203, and it's so it's upregulate microRNA 203, and this MIR 203 in turn suppress p63, and the p63 is known to suppress, known to cause keratinocyte proliferation. So then we Hongling mainly show that galectin-7 can activate JNK1, C-jun N-terminal kinase. This is kinase, and then we concluded that the galectin-7 through this pathway suppress keratinocyte proliferation. when it's downregulated, there's increase in proliferation. So they take care of the epidermal hyperplasia part. Then on the inflammation. So another microRNA that galectin-3 regulate, in this case, the suppression. Galectin-3 suppress microRNA 146A, and we show that MIR146A is a promoter of this MAP kinase pathway induced by IL-17 through IL-17 receptor, leading to IL-6, IL-8 production. These are two major cytokines contributing to psoriatic inflammation. So galectin-7 suppress these pathways by suppressing microRNA 146A. So we were able to conclude from two papers, downregulation of galectin-7 in keratinocytes may contribute to the heightened keratinocyte proliferation and the inflammatory response in psoriasis. Thus, approach to induce galectin-7 in skin may be developed as a new therapies for psoriasis. And we were lucky that by connectivity map analysis, this is CMAP. I'm not sure you're familiar with this, but that's okay. And it's very powerful in silico analysis. We found that statins, I'm sure you're familiar with statins, can induce galectin-7 expression in keratinocytes. Thus, we proposed that the statin may be employed for treatment of psoriasis. In the meantime, control this lipidemia. So we actually already proposed a clinical trial based on this, and statin alone, we don't think it will be effective enough, but together with other treatments, believe will benefit patients, not just for their psoriasis, but also for their dyslipidemia and for their metabolic issues. Okay, I will skip next few slides because I really want to get to the last part. So the first part of the slide I missed, I skipped is galectin-9 and galectin-12 is very interesting galectin that we discovered and has to do with lipid and it's expressed mainly in adipocytes. So I hope I will. Actually, I talked about this earlier during my earlier visit to慈济. Maybe share with you about our recent development with regard to this galectin, maybe in the future. So, because I want to talk about this final part. So, come back to the extracellular function. Although it can be easily demonstrated, I continue to ask whether exactly what they do, what galectin do in vivo, also in extracellular space. And although we know that they are able to engage cell surface glycoprotein, I gave you a number of examples of galectin-3, 7, and I skipped the galectin-9, 12. Inside the cell, they also can have an important role. They function by glycan independent, in a glycan independent fashion. So we've been asking do cytosolic galectins function by binding to glycans. So by glycan, I meant this slide I showed very early. So O-glycan, N-glycan, glycolipid, and proteoglycans. Inside the cell, I mentioned that they are all linked N-acetylglucosamine, but the glycans do not bind to these glycans. And the answer is galectins really not supposed to bind these glycans because this glycoprotein, glycolipid, the synthesis is initiated in the lumen of ER and the Golgi ER, and then they get modified in Golgi, and they can get outside the cell. They can be internalized and get into endosome and be digested, or they can recycle back to cell surface. But all this time, these glycoconjugates, glycoprotein, glycolipid, they're in the lumen of these organelles, ER, Golgi, post-Golgi vesicles, and endosomes, lysosomes. However, we didn't know. I wish I knew this earlier. I very much regret I didn't know about this actually can occur. So that is, they are disruptive materials that can disrupt endosomes or lysosomes, maybe other organelles. So this disrupt material, I'll come back, they are various kinds. They can be internalized, and this is in the process of endocytosis. In endocytosis, the glycoproteins, glycolipid, initially on the cell surface can be internalized, but they stay in the lumen, just like I mentioned in my earlier diagram. They are in the lumen of these vesicles, and glycans are outside. However, this disrupt material can cause damage to these vesicles, and then the glycoprotein, glycolipid can be exposed to cytosol. In cytosol, there are glycans like glycan 3, glycan 8. They can bind to these glycoconjugates very quickly, very readily. And then, in addition to binding, it's now known that they can induce downstream events, for example, autophagy. Yeah, this is a very interesting phenomenon or biological process. And so we were studying the infection of macrophages by Listeria. This is a very well-known intracellular pathogen, and they can be engulfed by macrophages, or they can invade the macrophage. But they are one kind of a disrupt material because they want to escape from phagosome and they cause damage to the phagosome. They can get outside the cells. So we show that glycans, this is time lapse, and glycan 3 can accumulate it quickly around. So this lysis process occur about 1 hour or so, and just around 1 hour, glycan 3 can be accumulated quickly around where Listeria was. Okay, this very important papers published. Very important paper published by a German group. That glycan 8 targets damaged vesicle for autophagy to defend cells against bacterial invasion. So they work with Salmonella, and we work with Listeria. And the Salmonella, like Listeria, can be engulfed or can be phagocytosed, but they can also, they want to escape and cause damage to the phagosomes. Then these authors published this paper published in Nature. This is 2012. There's already maybe close to 900 citations. I believe this will become the most highly cited papers in glycan field. So they show that 3, 8, and 9 can bind to the glycan in damaged phagosomes, and more importantly, as importantly, glycan 8 can recruit autophagy adapter NDP52 and induce autophagic activation. So we had to catch up, but very much inspired by the work, and then showed glycan 3 can function oppositely. That is, glycan 3 suppress this autophagy activation. So we published a paper a few years later, and this became editor's choice. Glycan 3 protects intracellular Listeria by suppressing the autophagic activation or the autophagy response in a host and glycan dependent manner. So this is glycan 3 bind to host and glycan, and 3 and 8 antagonize each other. Another example is with H. pylori infection, but I need to only go over to the final conclusion slide. So H. pylori, which you are familiar with, cause peptic ulcer. They are not intracellular bacteria, but they can generate produce virulent factor, for example, VAC-A. So vacuolating cytotoxin A cause damage inside the cell, and we show that VAC-A can cause damage, lysosome to be damaged. Then glycan 8, just like in the case of Listeria salmonella, can induce bind to the to damaged lysosome, bind to glycans in the damaged lysosome and induce autophagy. And we show further that the autophagic activation can lead to generation of reactive oxygen species and cause additional damage of lysosome. So these are important because there are many substances that cause endolysosome damage. Intracellular pathogen or pathogen that produce core-forming proteins like VEC-A. Virus actually can also cause damage to endosome. Protein aggregates like those involved in neurological disorders. So my colleague Chen Yizhuang老师, she in collaboration with us showed that in the disease she's expert of Huntington disease. The problem of the disease, the causing substance is the disease causing substance is mutant Huntington. These aggregates. Then they show these aggregates can cause damage to lysosome, and then this is known to cause inflammasome activation and glycogen-3 potentiate this process through binding to damage lysosome, and this cause increased inflammation in microglia in the brain and cause neurodegeneration. So this is the protein aggregates, but also particulates. There's not many examples, but we believe that, for example, PM 2.5, PM 10 particulate matters, and in fact, the nanoparticles that you're now familiar with because of the vaccines, they can cause this kind of process and leading to some inflammatory response, which is I'm not sure is widely recognized. And finally, cellular stress, that the oxidative stress is predicted to cause endosome lysosome damage. So so I'm sharing with you a novel mechanism mediated by cytosolically exposed glycans. And this can lead to diverse cellular responses, autophagy, inflammasome activation, maybe apoptosis, and this can lead to innate immunity in the case of infection, inflammatory responses, and this is associated with diseases. So I then come back to this slide. So outside the cell, we still do not know exactly what they do. They must be doing something important. But inside the cell, I share with you, they can function in glycan independent fashion. But also very exciting new recent development is they can engage intracellular glycan dependent glycan sensing responses. So I think I'm already exceeding almost. Actually, yeah, my time is up. So again, thank you for your invitation, and thank you for your time.
Just raise your hand and ask. Anyone want to share your opinion? Maybe I have a question for Professor Liu.
Okay.
Because you're talking about the gelatin-3 with the gelatin-8, and more like the antagonize each other in some tissue or cell. Is that for every kinds of the cell or tissues? Because it's very interesting. Because Professor Liu?
Yes. Yeah. So yeah, very good question. So I must first clarify. We look at the Listeria, and then the other group look at Salmonella, and we didn't look at gelatin-8 in relationship to Salmonella in our system. So, but we believe also from other studies, this gelatin-8 is in promoting autophagic activation. Gelatin-3 in other systems as well is suppress autophagic activation. So I believe this can be generalized in this endosome lysosome damage process. So yes, I think this can maybe generalizable, which is what something we were interested in doing, very interesting in the first place. I should mention that they bind glycan-3 and 8 bind to different set of glycans. Maybe there's some overlap, but they bind different set, and they do not inhibit each other in terms of binding to glycan. So it must be in the autophagic activation process. Could be so gelatin-3 is not known to interact with NDP52. individual. So it's through other mechanisms it suppress autophagy. Glycan 8 promote autophagy through binding to NDP52. Yeah.
Okay, thanks. Is that the Chen?
Yeah.
Raise your hand. Okay.
Fudong,
Yeah.
I have a question. So very similar to the previous question. So actually you just mentioned that galectin 3 and A, they do not interfere their binding to glycan.
Right.
We're assuming that when you have
not antagonize each other.
Okay. So when you have a damaged endosome, that so both of them will bind to the damaged endosome,
Right,
expose glycan at the same time.
Yes, right.
Oh,
Yeah,
okay.
Right. However, but through a super resolution SIM, we found they bind to different microdomain.
I see.
So
I
it's
see.
really very interesting. So
it's the data part how they engage with autophagy.
Right. Yeah.
That differentiate their function later.
Right. Yeah.
Okay.
We still have quite a bit to learn there. Yeah.
So did your team find out that like different stimulation, different damage, induce endosome damage will lead to different accumulation? Like, in some cases you will have more galectin 3, and in some cases you have more galectin A, or normally you will find equal amount.
So yes, not exactly to that extent, but we use the photosensitizer and then illuminate. This is the system developed by Yang Weiyuan, and so we modify a little bit. So we cause damage to endosome. He caused damage to lysosome. So the endosome, then we find the 3 and 8 really behave very differently. And so that's another system. We hope to answer your question more in more detail is H. pylori. So the study was galectin 8, but we also have galectin 3. Already have quite a bit of data. Have not published yet, but there I hope to compare 3 and 8, and answer the question you addressed. And again, also see whether they antagonize each other or not, and also the timing of their binding to the glycan. I should mention in the photosensitizer case, galectin 8 bind very, very quickly, like within a second.
Oh,
we shine a light and it's there. And 3 take about a minute or so.
I see. Okay.
Yeah.
Oh, okay. Thank you.
Yeah, thank you for the question.
Professor, I have a question.
Oh, okay. Yeah. Just ask.
Okay.
Maybe just introduce yourself a little bit.
Okay. Professor, my name is Ayo. I'm a PhD student year 2 here in Cincy.
Hi.
Okay, sir.
Yes.
So my question is this. First, I want to ask about how to solve the problem of the specific binding of the glycan binding proteins, because since there is a problem of them binding to different kinds of ligands. So how do we circumvent this event? That's my first question.
Yeah.
Like, how do we actually ensure that this does not happen when we begin to investigate any of the glycoproteins?
Can you answer this first?
Okay, sir.
So very, very important question. I'm very, very happy that you asked this question. So I think it's so 2 aspects. One is we just need to be aware of this issue. And for example, in the literature, including what we published earlier, it's very common that one of the galectins is linked to one glycoprotein. Then the rest of the paper or rest of series paper is focusing on that interaction, and which more and more clear is that may not be the case. So typical example is galectin X bind to a CD something, and then show that there's a response. And then conclusion is through binding to CD something, galectin X induce that signal, right? And then which is then later on find that this may not be the case. So just first be aware that we call the almost like a multivariant kind of situation, aware, and then the second aspect is maybe these glycan binding proteins are really different from the cytokines of those their function might just involve binding to many partners at the same time. For example, intracellularly, I talk about this damaged endosome. I didn't say that which glycoprotein they bind to. I believe they bind to quite a few. The glycan 8 bind to a set, but glycan 3 bind to another set, and they actually totally separate. But in that set, there might be more than one glycoprotein. So some cases you really do not need to know the specificity. Mainly glycans as a group suited to serve as anchor, and for glycan to engage glycan, right? So that way we don't need to solve, just need to be aware. Also need to be aware is extracellularly. Then what you need to solve is extracellularly. So what are you going to do? I believe it's very challenging. That's why after so many papers, I still put a question mark there. That outside cell, we need to know. We need to work with animal models and find out, say, one glycan coming out of macrophages, where they go to. They go to neighboring endothelial cells or epithelial cells or T cells, and then in terms of what cells get activated. So this really requires some kind of maybe new tools or the combination of many tools to dissect this, right? You see what I'm saying? Yeah. So we need to really go in vivo, in animal, and then look where glycans are. Might not need to know which glycoconjugates glycoprotein bind to, but mainly figure out where they go to. That's how I see that as a first step. That is already challenging enough. And then which cells are activated. Then we can say, yes, they're doing something outside the cell. Yeah. The mere presence of these glycans may not be the answer. So you find it's in the serum does not mean it work outside the cell. Yeah. I hope it's clear. I'm sorry. So your next question?
Okay, thank you, sir. My next question is, what role does, maybe because you've said that, okay, the gelatins, they play intermittent roles together, but in ubiquitination, which is also a process of maybe degrading proteins as well, have you worked on anything that has to do with that? Maybe is there an interaction or a link with that?
We don't work with that directly, but other researchers have published paper also has to do with endosome damage or lysosome damage, and they start to put the ubiquitination there, and in a kind of a pretty complicated scheme. So I think all these different processes probably interwave. Eventually we need to sort those out as well. So no, we have not looked at that directly, but the other researchers are looking at ubiquitination also, because this also can be maybe more ubiquitination also occurs when proteins are degraded in lysosome. So these are clearly somehow related, but we have not addressed that directly yet.
Okay, sir. Then my last question has to do with autophagy, because yeah, I'm interested in autophagy. I understand that for autophagy flux, it means that like autophagy process has been completed. So for the galactin 3 and 8, because you said they don't necessarily inhibit each other, does it mean that do they enhance autophagy flux, or like for galactin 3, does it suppress it or enhance it? Because one of the paper that you mentioned, I think there is like 2 different roles between the 3 of them. So
Right.
yeah, I just want to be clear, maybe which one favors autophagy flux, or does it have to do maybe in a disease condition? I mean, does it relate to that would determine maybe autophagy is actually activated or is downregulated?
Yeah, so it's really still we don't know we don't have answer to all those. It's still pretty early in this area of research. But mainly what we know is glycan 8. Only among 8, 3, 8, and 9, only 8 engage NDP52. So it's generally accepted that when in NDP52 is engaged, then there's autophagy activation. So that's all we know. And the glycan 3 does not bind to NDP52. So through what mechanism it suppresses autophagy, we do not know. So still lots of work need to be done. Yeah, very good question. Thank you.
Okay, have anybody have do you have a question? No. Okay. So maybe we just have a break because I think the Professor Liu just give us a wonderful talk for 1 hour. So just give the warm hand for the Professor Liu, then we will have the break. Okay. Thank you, Professor Liu.
Thank you.
所以那个刘院士，我们休息一下，您也辛苦了。
那
我们待会的这个学生的给一个那个 short talk 的时间，应该是在3点几分？3点05分哈，所以我想还有10来分钟，请那个院士您休息一下，然后再
我还是在线上吗？
是，您就把那个camera 关一下，声音关一下，休息一下。
OK，好，
谢谢您，谢谢。Okay, right now. 林浩然林同学，好，那他的指导教授是做寄生虫学研究的，好，那所以就是我们想说也是让院士来指导一下，听听看他们现在做的情况以及他们的方向。好，那我把那个麦克风交给我们的林同学。
院士好，各位同学好。那我是医科所的林浩然，然后我的指导教授是彭思怡老师。那我今天要跟院士报告的题目是，我们现在做的题目是那个我们想要利用这个 Syndrome B 五位子移素去治疗慢生血吸虫感染所导致的一些 organ injuries。那个慢生血吸虫它是一种寄生虫疾病啊。那如果未经治疗的话，它其实可以在人体里面存活5到10年，甚至更高。那在这个过程中，它会一直产生这个虫卵。正常来说，虫卵会经过我们肠胃道，然后就沿着 stool 就排出去体外。可是有一大个部分呢，它其实会 through 我们的 circulation， 然后进入到不同的组织上面，然后并且造成一些器官的病变。那最主要的几个器官都是包括那个肝脏、肺部还有肠胃道。那它们虫卵会在这个 liver 上面就是卡住，然后呢导致一些免疫的现象，然后有一些发炎，然后最终会导致一个 liver fibrosis。那又因为那个肝脏其实从生理学上或解剖学上，它其实跟很多不同的器官也有所连接，所以呢这个 liver fibrosis 其实也会导致一些其他器官的病变啊，譬如像一些脑部的病变或是一些心脏啊的方面的问题。好，那目前临床上治疗这个血吸虫呢，主要是利用 Praziquantel。Praziquantel 可以针对不同 species 的血吸虫疾病去做一个治疗。可是呢，它的一些 limitation 呢，就包括说它没有办法，虽然说它可以有效的去杀死这个虫体，可是它没有办法去排除掉本身在身体里面的一些虫卵，或是恢复本身的一些 organ injuries 啊，譬如像 liver fibrosis， 目前临床上好像也是利用这个 supportive 的 therapy 啊，所以是没有办法去恢复这些的 injuries。那另外就是说，在一些比较盛行这个疾病的一个地方呢，已经开始出现一些 resistant 啊，所以对于这个血吸虫病的治疗呢，就开始需要一些新的一个药物啊。那我们的研究呢，是利用一个从五味子果实里面萃取出来的一个 Syndrome B 五味子胰素去做一个治疗。那五味子胰素呢，在临床上就是用在人体或是在动物模式上面，都已经被证明说有很好的一个 liver 的 protection。那在不同的文献里面也有呈现说啊，这个五味子胰素可以针对不同的 liver injuries 去有一些不同的功效啊，譬如像发炎啊、抗氧化或是 apoptosis 等等。那所以呢，我们这边的研究呢，就是想要去利用这个五味子胰素去治疗血吸虫的疾病。那我们目前得到的结果就是说，五味子胰素无论在体外还是体内啊，也都是可以去破坏雄性成虫的一个表面。那因为它们雄虫母虫要进行一个产卵的动作，是需要 surface 的 interaction 啊，去进行一个交配。那雄虫的 surface 被破坏以后啊，我们发现虫卵数就是它们产出的一个虫卵数是有所减少的。那在体内实验里面也发现啊 ，organ 里面的一个 egg burden 跟一个 stool 里面的一些虫卵数也是有所减少的。那因为它没有办法完全取代掉 Praziquantel， 因为它没有杀虫的功效。那所以呢，我们就伴随着临床上这个 Praziquantel 啊，一起去治疗这个少鼠的一个模式。那我们发现啊 ，Praziquantel 偶和这个五味子一起去使用，针对于不同的一个 organ injuries， 都是有比 Praziquantel 这个单独治疗是有来得好的。然后另外就是不知道为什么这个 PPT 被挡住了，本身应该是有一张图，它是一个 survival。这个两个耦合一起使用的话，那个 survival 是有被 prolong 掉，就是说它有延长少数的一个存活率。那所以未来呢，我们是希望去继续探讨说啊， 因为我们已知这个无位质移输是可以对于血吸虫是有一个很好的一个作用。那我们未来就是想要继续去探讨它的机制啊，譬如像，因为以前的 report 是已经发现说 liver fibrosis 是跟一个 step free 的一个 signaling pathway 是很有相关的啊，所以我们在体内或体外也会进行类似的一个研究，想要去找到它的机制。那又因为那个 liver fibrosis 主要是跟那个 hepatic stellate cell 肝脏性状细胞是有相关的，我们也会利用这个 hepatic stellate cell 的 cell line 去进行一个细胞的研究啊。那这是我的报告啊，谢谢。
好，谢谢。那我想问一下，就是因为这个有2个部分，一个是说是它就是抑制这个虫，那另外是 injury， 对，所以这2个不见得是一样的事情。那就是如果说是刚刚讲说是因为那个，因为有些药即使它可以把这虫去掉，但是那个 injury 造成的 injury 还是没有办法恢复，那所以你又有新的药来治疗这些 injury， 我想这是一个。第二的话，但是后来又好像会变成说，它就本身就是对这个驱虫就有帮助这样子。这个当然也许就是中草药的强项，就它可以是 the complex formulation， 因此它可以有2个功能，那这样当然很好。对，但是这个的话，是不是能够你们有没有什么想法这是可以分开来讨论的？对，所以要不然的话可能会造成一些 confusion， 就是它到底是功能是在抑制那个还是回修护这个 injury 这样子的一个。对
啊，谢谢院士。那其实我们一开始是想要把这个中草药的成分是直接取代，就是我们一开始的研究目的是想要探讨说它能不能直接取代掉临床上的，因为它已经有 resistant 了。那如果能直接取代就更好。对，那所以我们前面的研究其实是除了有那个发现它有这个破坏虫体表面以外，我们也是有发现说它本身，这是我们一开始的实验室是单纯只用它去做治疗，然后其他什么药物都没有加，也是有发现那个 liver 的 fibrosis 是有所恢复的。那我们另外也有进行那个以那个
我稍微打断啊，你说你 liver fibrosis 有恢复，言下之意就是那个是这些已经用药物处理过，它还没有完全恢复呢，还说是你是讲说是 liver injury associated with 这个 infection 也没有，所以这应该是2个，一个是虫的量少了，一个是 liver fibrosis 少了这样子，还说是它是这个，所以我觉得就是这方面我听起来有点不是那么清楚，我想主要是这样。对
啊，好，谢谢。我们也有进行，我们除了那个虫体，我们也是想要排除掉这个关系，所以我们也有进行一个以 CCL4 去诱导。对，对。那从这个模式，我们也是有看到
，OK，
是有所恢复的。
对，那就很好，那就清楚了，就说是它可能有2种不同功能。那这的话就是这中草药就可能有是可能，西药的话就是会变成，也许它就是驱虫的。也许它是抑制 fibrosis， 不是说2个，就比较难说是看到2个效果。那这边说2个都看到，那也许就变成，对不起，不是，也许就变成说是不同的 component， 对不对？所以这边的话，以后也许就是要说是不是同样的 component， 一个是驱虫的，一个是 恢复 liver injury 的这样。对，社会稍微复杂一些。那我刚刚另外一个问题，也就是想说是知道说，是针对，这是 focus 在 fibrosis 上面，是 focus，或是我想再请教一下，就是它的 injury 最重要的 injury 是什么？是不是 fibrosis 呢？还是如果你
是的，它最重要的是 liver fibrosis。OK， 还有其他就像 non fibrosis。
也许就值得。就是那个是一个 focus， 然后那个话就像你已经做到就 CCL4， 也许有不同的其他的 model， 来看这个。那边有一个另外的这个extra benefit， 就说是事实上你会 develop 一个对 liver fibrosis 的药，而不是说跟着虫就没有 不见得有关系，但这也是好事情了，就是你这同时，但这个中草药在 liver fibrosis 有效，过去知道吗？已经知道了。
是的，是的，嗯，
知道，对。那那知道是什么 component 还是 ingredient 还是不知道？
就是它本身是一个果实啦。那在中医学上，它是直接就是服用这个果实。
我知道。
那所以我觉得五味子语素就是去找出啊，它究竟是哪一个 component 会比较有用。那从文献里面来看，好像是我现在做的这个五味子语素是比较是最，它是最 active 的一个成分。
是，对。那我想这个，如果说是要看它的机制的话，是会想说是去看那个 model fibrosis， 还是用那个 schistosome 这个 model 来看，就看它的 liver fibrosis 机制怎么 benefit 到那个。
那因为以前有人研究过，就是以其他 model 啊，比如像注射那个 TGF beta 啊，这是走这个 SMAP 的 signaling pathway， 就是那个五味子语素可以抑制这一条 pathway。因为在这个 schistosome 所引起的 liver fibrosis 啊，也是有被证明过，它其实有被证明说可以诱导这个 SMAP dependent 的，同时也有 SMAP independent 的。那我们就是想要知道啊，这个五味子语素在这个 model 里面，它究竟是针对哪一条路径？
是 ，OK。好，
指导教授有没有想要请教院士的部分？应该还好。好的，那如果院士这边没有进一步想要再讨论的部分的话，那我们是不是就 move on the next speaker？ 好，那我们下一位是群民吗 ？OK， 好，
谢谢。可是那就
那我们下一位是我们花莲慈济医院的张群民张医师哈，他是一般外科的医师。好，那我就请他来跟院士分享一下他现在在做的一些研究的部分。好，请。这是新的吗？新的放在桌面吧。
对 ，yes， 对，这才是新的。好，那刘院士好，那刘院士我是张群民，那跟刘院士报告就是今天要报的题目是就是 interleukin 10的 antagonist 的一些 peptide 对 breast cancer 到底有没有什么样的效果这样子。那其实我们知道说哈，因为 interleukin 10它在人体上最主要是有一个 anti-inflammatory 的一个 response， 那透过一些机转，比如说透过这个 MPK 这个机转，然后就是导致说对这个 anti-inflammation 的一些变化这样子。那目前在临床上有发现说，对interleukin 10它在于因为它对于immune 这边，它对 cancer 这边其实有两个，好像就是有些是看到 anti-inflammation， 有些是看到 anti-tumor， 有些是看到 pro-tumor 啊，所以它其实它的角色有一点还没有那么的一个清楚。比如说像这边的话，它会活化 CD8 的 T cell， 然后产生 interferon gamma 的增加，然后对于细胞有一些毒性cytotoxicity 的一些现象。那同时它interleukin 10它也有的路径是会去抑制 macrophage。那因为 macrophage 它这边底下其实像 macrophage 后面有这些 interleukin 12啊，然后活化 T helper 1 cell， 然后会增加 interferon alpha。那这条路径interleukin 10又会去把它抑制掉，所以其实那另外的路径包括说它也会去像这个抑制这一条dendritic cell， 它会还有 T helper 7这些cell 会去促进细胞的一些抑制，那它又把它抑制掉，所以变成说它其实它的角色是 either 是促进细胞的促进 tumor 的抑制 tumor 的 progression 跟或是促进 tumor 的 progression， 它好像两条路径都会有看到。哦，那在一些 paper review 起来看到说，好像的确就是在 paper 上看到说，真的是有些就是 anti-tumor 的反应，有些是 pro-tumor 的反应，那有些是并没有很明确的一个定位的角色。那所以说，目前这边跟大学老师合作，就是说想看看说interleukin 10 level 在 breast cancer 的一些 specimen 上啊，还有病人的血清上，的关系性是怎么样？那也看看说它的 interleukin 10到底在breast cancer 它的角色，我们这边可不可以看做到了结果是怎么样？那再看看说使用跟另外的老师合作的发展的 interleukin 10的 antagonist 的 peptide，或者是有没有是 antagonist 的效果，是 agonist 的效果这样子。那目前的话，大概会预计要进行的就是说先分析说 interleukin 10在 breast cancer 上的一些 effect。那我们也是传统的，就先看这些 proliferation 啊、migration 啊，或是 EMT 的一些变化啊，做病理的染色跟 cDNA microarray 的分析，还有或是做之后再做PCR 的分析。那最主要的话之后会做到cell line 之后会再做到动物上，那看看说到底是不是说interleukin 10它在动物上我们看到的一些表现会是怎么样子。那目前我们大概用的就是些我们市面上常看到的三个 cell line 啊，就是 triple negative 的，然后还有ER positive 的跟 HER2 positive 的乳癌的3株细胞株来做，还有病人的 primary cancer cell。那目前也是在在开始培养说，病人的那个 primary cancer cell 啊，这是一个 HER2 positive，就是荷尔蒙阳性 type 的一个 primary cancer cell。那目前有看到了一些刚开始做只看到说，比如说interleukin 10对于 cell 的 proliferation 好像我们在用 diversity-1 去染色去测它的cell 的 proliferation， 好像并没有看到说 interleukin 10在对于 breast cancer cell 的 proliferation 有任何的影响这样子。那随着时间的增加，好像它对于这个 breast cancer 是没有什么样的影响啊，至少在 proliferation 这一块看到是这样。那在 migration 的话，不过似乎在 migration 上就看到说，的确是它好像会促进感觉啦，就是说跟 control 比起来的话，好像浓度比较高的interleukin 10在这个ER 这是 HER2 positive 的 breast cancer， 好像它会有一些促进 migration 的一些效果这样子。因为 migration 也是一些 EMT 的测一些 EMT 的 marker 嘛，那所以我们就看一些 EMT 的 marker， 看到好像的确是看到 eclahirin 就在比较高浓度的那个interleukin 10下，那个它的 eclahirin 就变得比较少。对，那我们也有做说一些初步的一些 breast cancer 的组织染色，那看跟 normal cancer 来比，这3组不同的病人，因为目前样本数很少啦，但是好像没有看到明显的一些关系，就在里面好像normal 跟 breast cancer 比起来，好像这个好像有比较多啊，这个看起来又好像第2个病人的 sample 上看起来又好像不明显，那第3个看起来好像也不明显，所以其实因为可能样本也很少，所以目前它的关系性是怎么样，其实我们也还没有很明确。是以上就是目前的一些进度跟刘院士分享。
好，那个这里的话，就说是在不同的 cancer 有不同的角色，有像事实上也是不跟我刚才演讲不完全一样，但是就是会有事实上这2天还跟我生医所的研究员谈了一些，他有一个他的有兴趣的一个 molecule， 在有某些 cancer 是 tumor suppressor， 某些 cancer 是 oncogenic， 那我们也是同样的 conclusion， 就是有时候可能是就是那个 system， 然后真的是蛮 challenge 的，要把它 dissect 出来，对。所以，但是这个是可能的，但是会有个 challenge， 就是到做出来之后，就是比如说 breast cancer 就完全没有，应该有人已经做了嘛，是跟 IL-10的关系，对不对？那就会比较难，说是 one way the other， 就说是你的最后的发现的新的是什么，对。
是，
那是 breast cancer 上面也有说，有人是说 pro， 有人是说是 anti， 就是 breast cancer 上面，对，所以这边的话就会比较 challenging， 我有这感觉。 那跟 IL-10 做的人也是非常多了。对，我也问他，我先问一件事情，然后再有一个想法。一个是你说是那个peptide 当 antagonist 的那个是什么样子的 design？就是 IL-10 的一部分是？
对，其实老师就是说，我们就是用也是用电脑去模拟，然后去算，就是电脑去 docking 看看说，用 IL-10 的 receptor， 然后去做 docking 一个药物，然后来做模拟看能不能把它做。
你说那个是一个 peptide，那是一个 peptide， 会想做一个 peptide。
目前是这样子，嘿。
OK， 对，那个药可能不太好，会蛮不好用的。因为 peptide 通常不是 stable， 而且要变成 d amino acid 啦，这个过程有点复杂。对，
是。
对。
好啊，那老师有没有建议？建议用什么比较好？
我感觉这个方向有点 challenging。对，所以但是我不晓得你已经投入多少。我倒是有个建议，就是因为就是我们也很希望就过去那个慈济跟中医院就是已经几年来，就是大概最近3年不可能不止，就是我们新院长上任之后有很好的互动，然后有些经费是慈济 support 的，也是会跟中医院合作这些。那现在在 breast cancer 还没有，我想你知道呢，在 lung cancer 那一边，就是 Cancer Moonshot 里面发表的一些那个东西，那个是在 Cell 发表，看到非常多的这个有很多 information。breast cancer 还没有发表，但是也快了。我昨天才听到一些 update。那个不也是一样，跟那个 lung cancer 一样，发现的应该是会非常多的新的 information。对，所以我不晓得这个，当然我不能 speak for 这个团队，但是事实上是我主导的团队，就是我们这正则度的计划，我是总主持人，然后有2个分支，一个分支是 Cancer Moonshot， 然后他们4年来也非常好的成就，继续下去了。那我现在已经不是主持人，然后另外分支是 Microbiota， 成就也是非常好的结果，那还没有发表就是了。那昨天我还知道。那个就陈玉茹老师，他过去是化学所长，他在 breast cancer 他也已经有一些发现。对，所以我是会鼓励这种合作。当然我是说是我不能 speak for 他是不是愿意，我相信他看到他的 breast cancer 跟 lung cancer 一样，都是非常大的团队。你去看那个 Cell 那篇文章，不晓得多少个 co-author， 这是很好的事情了。我觉得这是现在的趋势，然后 co-corresponding 就好几个。对，我觉得都是好现象，就大家愿意合作。台大团队主要是，那我觉得这个 breast cancer，如果你有兴趣的话，我可以介绍，然后我相信他们也会非常欢迎。要在这一方面呢，是不是看到哪些东西，是不是继续 confirm 也可以，然后或者继续就下去机制，然后做的是当然是有合作方式才能够会有更有效果。对，我是这样想。那正好今天听到 breast cancer， 然后昨天听到陈玉茹所长讲他最近的进展，讲到 breast cancer， 所以我就想到说是不是这边可以帮忙我可以帮忙介绍。对，也许你已经知道那个团队了吧？陈玉茹老师、陈所长，还没有的话也都没有关系，我们就是然后有兴趣的话，当然这个是都不能勉强的。
好，太好了，感谢刘院士，谢谢。
也就是让我知道，那我就会做个 introduction， 他们会非常乐意，因为就是很需要这方面的这个 clinical 的专家来一起研发这样子。对，
好，谢谢刘院士，感恩。
好，谢谢。
好的，那我们接下来是对不起，李玉。
我再问一个问题啊，再问就是如果针对 IL-10 的话，你希望看到是 inhibit 它嘛，对不对？对，那就是有其他不同的技术来做这个，那这倒是就是有些方法就是让它没有这个 cytokine 大概不太合适，我刚想的是比较 intracellular 里面有些方法能够让它degrade 掉，比较容易 degrade， 所以就变成不产生。那这个我不太觉得是可行，因为它是会经过 IL-10会经过 ER 沟局讲出来，所以这个我刚才想的这个方法可能不太合适。总是有一个想法，就最近想法是因为现在 mRNA 非常受到重视，所以也就带动了这个 RNAi 这种技术，说不定会进展的很快。对，那我们实验室也是，过去实验室我们拿到一个 grant 来说是制作 glycan 8的 inhibitor， 那这我们也做了一阵子，看到一些，但是现在我们想法就是我们就不要去管这个什么 compound 了，我们就做 RNAi siRNA， 然后来当做药。那这个我们看的是 psoriasis， 我今天没有讲，但是 psoriasis 所以就是用皮肤，就是 transcutaneous 的。那这个相信短期内这个 RNAi 经过皮肤进去，应该是会有方法。对，所以那这边也是一样的 ，tumor 是不是可以用 siRNA 来做？那我target the glycan IL-4 IL-10。但这边我就又必须讲说是也是跟我提的 glycan 的领域也是有关系，就真的不知道它是就是要知道它从哪里来，对 IL-10 哪里在哪里作用，所以这个肯定要搞清楚才知道是怎么样 target 它，对，所以 OK 好，就先有些想法，
谢谢刘院士，
对一下 OK 好，谢谢。
我们接下来是那个吕玉英李同学的报告，好。
刘院士好，那各位老师、各位同学大家好。那我是慈济医院的临床心理师，我叫李玉英。那我的指导教授呢是慈济医院的神经科医师罗燕宇医师，这样。那我这个研究呢，主要是就是我今天要报告的是一个算是研究计划这样子。那主要是在于就是正常的老化当中，然后去探讨说多重共病症对于认知功能的一个影响。那其实我们也知道说，在世界各国都有一个老龄化的趋势，那台湾呢也不例外。那在老人的这个人口当中呢，就有发现到说，就是 dementia 跟多重的一个慢性病症呢，其实有比较高的一个盛行率。对，那在有失智症的人当中还有包含就是轻度认知障碍的人当中呢，他们患有共病症的比例也比一般人还要高。那患有一些血管性疾病，然后或者说肝硬化啦、气喘或者说是糖尿病的人他们认知退化的比例机会也比较高这样子。那如果说多重共病症的一个负担增加的话，那也有发现到说，在失智症的人当中如果多重共病症的负担增加，那他们的认知的退化也会比较明显。那过去有很多的研究，其实他们有指出来说，可能不同的研究，他们针对的不同的一些认知功能去做探讨。那多重共病症呢，在这些老化的过程当中，也会影响到认知的功能，这样。那因为失智症呢，它其实主要的症状呢，就是在认知退化的一个部分。那认知退化又会影响到后续的一些生活的一些功能。对，但是以目前来讲对于失智症其实还没有一个很有效的一个治疗的方式，所以我们想要去识别说，那是不是有些可能可以改变的一些因素？那共病症就是这些慢性疾病呢，是不是一个可以我们探讨的一个可以改变的因素这样子？那所以这个我们的研究目的呢，就是在探讨说，就是多重共病症的一个负担，然后在就是整个认知功能的一个影响。那因为过去的研究当中，它大部分是在探讨说单一一个疾病，或者说以疾病的数量，那可能对于某一些部分的认知功能的影响来做探讨。那我们是希望是纳入比较多的，比如可能包含就是数量的部分，多重共病症的数量，那也包含多重共病症的一个严重程度。对，因为疾病其实每一种疾病的严重程度可能会造成身体不一样的负担。对，那所以我们也会考量就是疾病的严重程度。 那在认知功能的部分呢，我们就比较完整性的去考量说，可能我们也包含了就是学习与记忆啦，或者说一些知觉动作的部分、执行功能的部分、注意力跟语言的能力，然后来探讨这些相关这样子。那因为在过去研究当中呢，针对疾病还有认知的能力呢，也有一些其他的影响因素，那包含年龄啊、性别、教育程度、BMI， 然后还有抽烟、喝酒的状态、血压跟一些糖尿病的一些指数的部分。那所以我们这个也会纳入做探讨。那我们的研究方法呢，是以一个 cross-sectional study 的一个方式，那会在2年间呢去招募200名，涉及于花莲地区的一个尚在执行志工任务的这些65岁以上的志工。那我们会以面对面的访谈方式，然后还有测试的部分去做这个检查，然后跟收集资料。那会请神经科医师，就是受过神经科医师训练的合格的护理师去收集人口学资料，那用那个 CISG 这个量表去评估多重共病症的一个负担。然后也会请合格的临床心理师呢，针对不一样的认知的功能去施测这些认知测验。那跟慈济医院这边去合作说，就是抽血取得这些血液的样本，然后再去做分析这样子。对，那我们希望说，就是在失智症之前呢，我们就已经先识别出来说，可能这些疾病对于认知功能的一个影响，那可以提供说未来可能在预防性的精准医学上面有更多的一些预防的一个策略。好，谢谢老师。
好，很好，谢谢。这边，我能想到就是在台湾的话，但是我一面听一面在多想，就发现也许不完全能够用到，就是台湾已经有些大的 bank， 台湾 Bio Bank， 那现在政府也有整合性平台，我觉得各个大医院，所以样本可能在这些 Bio Bank 里面已经有一些。但是是不是那边大部分是不是能够去能够要用可能要用合作才能 contact 到那个病人？用在中医院主导的台湾 Bio Bank 这样说的话，现在已经有15万、16万的那个收案了。然后都有genotyping，几乎都有genotyping，所以是 very powerful 的。然后这里面有哪一些有什么疾病，大概知道，因为中医院的是以健康人，不是健康人，社区民众为主，但社区民众就是都可能有一些diabetes 啦，三高可能都有，然后有些有不止一个，可能有2个，所以这些资料是一定可以拿得到。但另外的就是台湾 Bio Bank 是计划是有追踪，每3年5年会追踪，那就现在看经费已经有3万多追踪，所以在这个追踪的里面有一些也许写失智症的早期的话，可能他在问卷上面就会提到我现在有些问题，对，那这边也许初步的就可以看到是不是有些关联。那这个样本数非常多，就是你想想看，就是我们其实从追踪那边来看，那追踪的话，我们都知道它3年前、5年前那个时候，这个台湾 Bio 已经有10年的历史，还有2年就是最先的规划是做12年，那有这个10多有3万有追踪的，所以如果我们就是重点放在追踪的话，有3万，那这个它是有些什么有没有共病，那个也都知道，哎，那这个3万里面不晓得有多少会有这个失智的，也不见得会那么多，有失智也许不会参加，不会再回来被追踪，对，但是应该会有，尤其早期的话，说不定是这个可以找到一些关联，那是一个方法，已经是这个，也就是设立 Bio Bank 的目的，就希望研究学者能够来用，我们也是非常希望说能够用的人会更多这样子。 那个可以，你看你的 design 是很好了，就是而且在医院里面有心理医师、医生一起合作，我觉得很好。但是可以考虑，就是台湾 Biobank 或是整个整合型平台，也就希望大家能多用。没有用的话，这就没有这个效果。那台湾 Biobank 是我说做了10年，现在是收案已经有16万左右，追踪的已经有其中的3万4万在追踪，陆续还会再继续追踪下去。对，所以也许请你另外一个跟这个有关这个，我想实际也参与了，有 TPMI 是中研院生医所的郭佩恩所长，他的计划，他的更 ambitious， 他是100万个人，然后就是全部做genotyping，他现在收案数是33万左右，对，那里的话，那个资料健保的这个在那个医学中心的资料就是 health 的 data 都能够连在一起，对，都连在一起。那边很可能也是一个快的方法，但是我不太确定那边是不是已经开放，就是让 user 去连，这个我不完全确定。不过有兴趣的话，应该我们可以帮，就是很容易跟那个 TPMI， 他已经 engage 实际了，所以就是，如果您听到这个没有 TPMI， 有没有听过这个？还没有，对，那这个。对，这也感谢慈济也对这个有兴趣，就是几个大的医学中心都加入了，就是收案，帮忙收案，然后重点就是做那个一次的genotyping，对，现在30多万，30多万，对，还有距离到100万还有一段距离，但是很快，对，这2个可能性，就是请你们想一下，也许可以一方面帮忙，一方面就帮忙这些在国家型的这些计划这样子。收费就是 TPMI 像那个的话，就是如果资料已经释出的话，就是几乎是不需要收费的。那台湾 Biobank 目前的就是也是你们应该是不需要简体，就是资讯就是了，那几乎等于免费了，对。好，就是这些建议想法，没有其他的。
那个院士，我这边帮这位是心理师啦哈。
是。
那我补充一下，因为台湾 Biobank 的话，基本上就是用申请制的，那会从资料库里面捞。那譬如说你设定了一些 criteria 之后，包括你调出来，假定你有设定一个项目之后，然后可以捞出来是其他的共项的因子，那是需要经过中研院申请，因为我偶尔也会审这个中研院送过来的案子，对，是。那我比较好奇的是那个李心理师这边哈，因为你的那个认知测验是用 MMSE 吗？
不只是用 MMSE， 因为其实台湾我看到很多过去的研究是用 MMSE， 可是 MMSE 它其实是比较一个 rough 一点的 screen 的一个 test，对，那所以我会用各种不同的，就是很针对性的，比如说我就是针对 attention 的，然后针对 executive function 去做不一样的那个 test 这样子。
OK， 因为我这边会特别讲到说，因为我们一般在做一些 cognition task 的时候，通常就是会设在那个 scale 上面。那通常做几千人的时候，有时候我们几百人、几千人，有时候还是会有点担心的事情是，可能在某一个 scale 里面的 population number 是不足够的。那当在跑那个 statistics 的时候，就常常会有一点点不足，然后你就没有办法去看到变化，然后就又要再 new recruit 一些这个 subject， 所以这个大致上可能都会有一些这样的情形呐，所以会还蛮辛苦的这样子，好。
对，这边的话就是 TPMI 的话是好一些，就是说是他们应该是可以 contact 这个就是个案。TWB 的话，如果你真的看到 correlation， 我建议的是看 correlation， 只是说初步的看是不是有，就是等于说是一个 proof concept， 然后就看到说是不是有这些 correlation， 应该会看到，但是下面要去真正去看它的实质的程度，这些就会比较 challenge， 因为没有办法去 contact 那个。然后，但是就变成是初步的看到有这 correlation， 那这个是因为是都已经有的 bank， 所以真的不太需要花太多的 effort，就是能够看到这个，那这个也有它的意义在。那 TPMI 就不一样，就是它事实上你可以知道它的 identity， 那是谁，然后就看出那些是不是recruit 他们来做这个就是做刚刚讲的那个 testing 这样子，对，就可以事实上是可以做到。那边的话等于说这是 TPMI 的团队在帮忙收案的意思了。对，因为从那边很快就知道说哪些有结构，哪些有些什么病，然后有没有失智的一些资料都会有，对，然后可以 contact 这个个案，对，就是可以考虑一下。
好，谢谢院士提供的一些资源，然后谢谢老师，谢谢。
那就再麻烦我们李心理师再回去跟罗彦宇指导教授再讨论哈。那我们接下来是梁少峰梁同学，那他是我们这个转译医学博士学位学程的同学，那他的advisor 是我们的刘一军校长跟我们中研院的陈建章陈研究员哈，所以他现在应该人是在中研院这边，也会利用 Google Meet 的方式连结。那我们是少峰你已经上线了
Yes，
有听到吗？
有，
我
听到。
OK， 好，那你如果 OK 的话，就让我们大家知道一下哈，谢谢。
Okay, so can you see my screen now and my voice is clear?
Okay. Great.
Okay, so good afternoon, everyone, Professor Liu Fudong and every professor and students. My name is Liang Shaofeng. You can call me Jason. So I'm the third year of translational medicine PhD student. So going to briefly introduce my research. So the topic is the CAV 3.2 knockout mice exhibit social and fear memory deficits and increased self-grooming and impaired recognition, and investigated its possible underlying molecular mechanisms. So
I think we're losing your slide. Yeah. Somehow I see only very small screen. Yes. So you probably maybe it's from the慈济side. You can modify that. Right? I do not see the whole screen.
How about now?
No, it's the same. It's not, I think it's not from your side. It's from the慈济side. I didn't do anything. Now I just see all this attendance.
院士，你可以做的事情是点他的那个 screen，
这样子吗？好
，double click 之后看看会不会变成 full screen。
现在回来了。这样。Okay, thank you.
Jason, is that okay?
Yeah, okay.
Yes. Okay, so my advisor is Professor Ingrid Liu in慈济University, and Professor Chen Jianzhang in Academia Sinica. Okay, so we are focused in the gene named CAC-NA1H, the CAV 3.2. So this gene encode the T-type low voltage gated calcium channel. So as from the figure you can see, here this is the alpha-1H subunit is the main unit to make the calcium flow into the membrane. So once the calcium influx, it will activate several pathways such as activation of calcium dependent enzyme, neurotransmitter release, gene expression, and neuronal excitability. So from previous study, we showed that this gene is highly expressed in the hippocampus and also the layer 5 of cortex. So from Allen brain atlas, you can see from in situ hybridization showed in the hippocampus, they have amount of mRNA expression of CAV 3.2 and also the layer 5 of cortex. And from 2006, this group using the human genetic study showed the missense mutation in the CAC-NA1H was associated with the autism spectrum disorder. And from the figure A, it shows they identify the mutation site in the this channel, and also from the functional analysis shows this mutation significantly reduced the activation of the CAV 3.2 channel. So I will introduce about the autism spectrum disorder. So from this 2013, the DSM-5 from the American Physician Association, they determined the ASD have two major symptoms. The first is the deficit in social communication, and the second is repetitive behavior, and also restrict interest and resistance to change. And this condition is a long life condition. As for the epidemiology of the ASD, so the prevalence of ASD in US is 1 in 54 children, and in China is 1 in 143, and in Taiwan is 1 in 313. Although the number is quite low, but overall the number of ASD patients is increased every year. So currently there's no cure for ASD. So now their therapy strategy is using the behavioral intervention to let the patient learn the social skill, and also they can become independent in the future. And also the ASD patient also have some comorbidities such as depression, anxiety, epilepsy, or gastrointestinal problem. So the doctor will prescribe some medicine to make them relief from this problem. And from this paper, they show that the overall lifetime of cost of ASD patients is around 3 million in the USA. So it's a quite large number for each individual. So it's important to study about the ASD. So the research direction of ASD, from the previous study, they showed that there's a structural and functional change in both glutamatergic excitatory and the GABAergic inhibitory circuit in the ASD patient. So this EI imbalance also exists in several mouse model of ASD. And using optogenetic or chemogenetic to manipulate the EI transmission, it can, such as in the medial prefrontal cortex and hippocampus, it can reverse their abnormal behaviors. And in this paper, they show they use the clonazepam, which is a benzodiazepine drug, which is a positive allosteric modulator of GABA-A receptor. So it can reverse the behavioral deficit in the mouse model of ASD. So this is the drug we are going to use, and this is a small molecular drug. And in this figure, you can see this from the top view to see the GABA-A receptor. So you have 2 alpha, 2 beta, and 1 gamma. So the action of this drug is once the GABA and the clonazepam bind to the receptor, the structure will be more stable. So make the chloride influx, increase the frequency of chloride influx, so that the neuron will be silent. Okay. So we have the CAV 3.2 knockout mice, which was generated from the Dr. Chen Jianzhang, and from previous study we showed that there was an impaired behavior in the hippocampal dependent text, such as trace fear conditioning, passive avoidance, and also we found in the hippocampus they have in the late phase of long-term potentiation was impaired in the knockout mice. And in 2014, in this group, they showed that this knockout mice has exhibit the elevated anxiety-like behavior and impaired in the recognition memory. However, there's no report to study the relationship between the ASD-like behavior and the CAV 3.2 knockout mice. So I would like to study this part. So we are going to use the several behavioral tests to evaluate whether they have the ASD-like behavior. And also we're going to, once they have, then we'll use the clonazepam treatment to investigate whether manipulate this GABA transmission can reverse their phenotype or not, and also further to identify the mechanism. So this is the result. First, we use the 3-chamber social test to investigate their social interaction and the social novelty. So they have 3 trials in this test. So the first trial is habituation, that the subject makes freely explore in the chamber, and in the second trial, the stranger mice will put inside and with the container, so the mice can smell and see the mice. The subject max can see and smell the stranger. Yes, so usually the mice will spend more time in the stranger 1 chamber, and after 10 minutes, we'll put another stranger 2 into the other chamber. So normally the subject mice will like the new mice, so they will spend more time in the stranger 2 chamber. Okay, so these figures show the result of the social interaction. So in the white indicate the wild type mice, and the dark is indicate the knockout mice. So you can see the CV3.2 knockout mice did not have impaired in the social interaction. Even we give the clonazepam by the in IP injection, also did not affect their social interaction either in wild type or knockout. Okay. But in the social novelty, you can see the wild type have prevalence to interact with the stranger too, but the knockout mice did not show this phenomena. And once we inject the clonazepam 30 minutes before the test, this abnormal phenomena can be reversed. Yes. Okay, and for another behavior, we use the self-grooming to evaluate the repetitive behavior. So the first 10 minutes will be the, let me show you the video. Okay, 10 minutes will habituation, and the second minute we will record how many times they do in the self-grooming, so indicate the repetitive behavior. Okay, so here's the result. In the knockout mice, they showed have increased self-grooming time. And through the clonazepam treatment, you can see it can significantly reduce their self-grooming time, indicate this drug can reverse their elevated self-grooming. Yes. And sorry for the limited time, so I didn't show my molecular experiment result, but we found that the one of GABA A receptor subunit was reduced in the hippocampus, and in the future I would like to use the immunofluorescence staining to reconfirm this phenomena. Yes, so this is my talk today, so I would like to receive the suggestions and the questions. Yes.
Okay, thank you. Very nice. So I'm very glad to meet students in this translation medicine degree program.
Yeah.
It's so there's already one from慈济graduated, right? I think she's cardiologist. I met her just about a couple weeks ago. She actually still spending time in collaborating with her mentor, Meng Ziqin 老师.
Yes.
Yeah, so I met her first at慈济.
Dr. Li Yuezhang, right?
Right, yeah. Yes. I also know that Zhang Dongxing is now directing program director of this program. So how many other students in addition to you currently in
one more fresh
okay, good.
first year student, one more.
All right. Yeah, I'd like to see more.
Yeah.
So yeah, very nice. So you mentioned that so this calcium channel knockout has some effect on GABA receptor or something in the molecular study. So that seems to be challenging to me. That is, how do you explain the phenomenon?
Yeah, because we saw the drug injection has effect, so that we are trying to think maybe these knockout mice have impaired in the GABA maybe transmission pathway molecule. No matter is the production of GABA enzyme or the receptor was reduced. So we use the qPCR and the Western blot to clarify whether which molecule was changed. So now till now we found that the GABA A receptor of beta subunit was reduced. So this one here, one of the GABA B receptor is beta 2 receptor beta 2 subunit was reduced. So why beta 2? Because we choose this is the beta 2 is composed around 50% of the GABA A receptor in the CNS in the central nervous system. So maybe this abundant, maybe so that we are focused. First we try to see the most abundant GABA A receptor, the subtype. Yes, and we found this beta 2 is reduced in the hippocampus. Yeah.
Okay. This is a new finding. Let's not reported before in terms of the calcium channel and this one.
Yeah.
Very nice. And what's your why do you think is the mechanism how calcium channel is interacting with this receptor?
Yeah, I think this is a good question that I need to how to say trace why this unit was downregulated, but because of the knockout of this type of calcium channel. I think this is the direction I need to find out. Yes. So till now I do not have the answer for these questions. But
do you plan to do any kind of screening, like high throughput or RNA-seq, those kind of things during the trend, and that can be easily done?
Yes.
Professor Chen Jian老师.
Okay, maybe I will discuss with him later.
Right, this currently a very powerful approach, and maybe sometimes I feel it's overused, but very powerful, and it gives you some idea of what happens when you do not have this channel. Right.
Yeah. Okay.
Very interesting.
Thank you, Professor Liu Hudong.
Good luck. Good luck.
Okay, thank you. Thank you, everyone.
Graduate soon and attract more students to this program.
Okay, thank you.
好，那我们下一个是最后一位梁廷瑞梁同学。好，那他现在也在现场了。那我们是不是就把麦克风交给我们的梁同学？
有听到声音吗？
有。
好，那院士好，然后老师同学大家好。那我是今天的报告者梁廷瑞。那我是慈济大学药理暨毒理研究所博士班的同学。那我的指导老师就是有药理学科的刘朝龙老师跟那个彭思怡老师这样。好，那我今天主要讲这个题目就是这个antimicrobial fusion protein， 然后我们称它叫做 lysostafin P3 这样。好，那首先我们先，因为它是一个 antimicrobial protein， 所以我们先介绍说这个它要针对的细菌。好，那其中一个就是第一个就是这个 Staphylococcus aureus， 好，那它是金黄色葡萄球菌，那它是一个感染式阳性的球菌这样。好，那虽然说它在我们的比如说上呼吸道或者是说我们身体的某些部位它是属于这个 normal flora， 可能会遇到，但是如果它位移到不同的位置，或者说我们有一些伤口，然后又或者是有些手术导致这个细菌的入侵，好，那我们的免疫力又就是没有办法有效的抵抗它的时候，那可能就会造成一些刺激性感染。那除此之外，比较严重的问题就是说，金黄色葡萄球菌呢，它有一些 抗药性菌株，像是那个 MRSA 或者是临床上有一些 ORSA 的等问题。好，那到未来可能会面临这个无药可用的这个窘境。好，那所以我们第一个针对这个金黄色葡萄球菌这种革兰氏阳性细菌的策略呢，我们是利用这个 lysostaffin 这个 protein。那 lysostaffin 这个 protein，它是一个螯合锌离子的peptides。好，那它也是从它是从另外一只 Staphylococcus simulant 中纯化出来的。好，那就一开始是从这只细菌找到的。好，那它的功能是针对这个它的功能是针对这个细胞壁上面，然后这个有5个 glycine， 对，5个glycine 的这个bridge， 然后进行就是它会有那个 peptides 的效果，就把它切断，那就可以有效去破坏细菌。好，那这个 protein 它有2个 domain。好，那前面这个图上这个比较绿色蓝色这个 domain， 它是具有peptides 活性的 domain， 那后面这个橘色的 domain 呢，则是这个它是具有 binding 的这个效果。好，所以呢，我们这一次实验会截取这前面这个部分。OK， 好，那另外一个我们要针对的细菌是这个 Acinetobacter baumannii， 那就是包氏不动杆菌。那这一支是革兰氏阴性菌，那并且它是造成一些刺激性感染的或是院内感染的细菌。好，那它主要就是感染一些就是说它会寄生，不是寄生，就是它会附着在一些皮肤啊或者是伤口之类的，然后这个大自然都有。好，那如果病人的免疫力缺失或低下的时候，那就容易造成一些更严重的感染。那除此之外，也是因为这些细菌可能有multiple drug resistant 的问题。好，那针对这支细菌，我们这里采取的策略是利用这个lysab2 的这个 protein 的其中一个片段。好，那 lysab2 它一开始是从这个包氏不动杆菌的phage AB2 中去找到它的那个endolysin。那这个 endolysin 确实它这种 endolysin 是这个phage 本身的 cycle， 那它最后要破菌时候自己会产生的一种分解细菌细胞壁的一种策略。好，那其中根据过去的研究发现，这 lysab2 的氨基酸的113到145这一段那它其实本身就有这个抗菌的效果。那并且就是之前他们做实验，就是发现他们稍微在上面改了一些氨基酸之后，那效果会更佳，那并且会减少这个溶血的效力。那其中 P3 的效果是最好的，所以我就把 P3 的这一段，然后拿来使用。那我们的策略就是把那个 lysostaffin 的前面的部分，然后跟这个 lysab2 后面的那个 amino acid 的部分，然后fusion 在一起。那 fusion 在一起就用 cloning 的技术。那 fusion 之后呢，我们就会进行一些纯化。那其实在这个地方有发现一些问题就是说它在进行那个 nickel current 纯化的时候，那它的吸附力是还蛮强的，甚至是最后还是要利用就是整个把离子全部都洗掉的方式才有办法把它整个都取出来，那并且在浓缩上面也有一些比较困难的部分。好，那但是呢，我们试着就是说，那既然 protein 已经有出现，那并且有拿到一些产品，那我们就做一些测试。那我们主要测试就是先针对这2只细菌。那左边的图就是这个那个 AB 菌的部分，那右边的图就是金黄色葡萄球菌。那可以发现就是我们是在水的环境下做这种抗菌试验，那可以发现它的抗菌效果都还可以这样。好，那之后我们会做对于整个流程会做一些调整，包含就是说这个纯化上面跟浓缩上面的话的一些条件的去调整。好，再来就是可能会试着使用不同的环境之下去测试那个抗菌的效果。好，那 如果前面都没有问题的话，那也许会往后去试着说，那如果它会不会有一些这个溶血的效果，或者是说它会对 plasma 有抗性？那这些问题如果都幸运的排除的话，那可能也许有机会进到这个动物实验的部分。好，那我今天的报告就到这里，那谢谢大家。
好，谢谢，很好。那个，事实上今天我学到新的，就是不少的 bacteria 会有 antimicrobial peptides、antimicrobial 的 protein。不就是你已经有2个例子，但是是不少或者说是大多数的 bacteria 都会有 antimicrobial 的功能。
你是说这个例子吗？还是说
没有，
目前
就是你提了2个例子嘛，但是其实2个不同的 bacteria， 但是其他的 bacteria，我随便讲，我今天提到的 Listeria、Salmonella 都有这个 antimicrobial 的 protein peptide。有都找得到这样子。
像以 peptide 部分是现在在网络上已经有 antimicrobial 的资料库了。然后然后一些 protein 大部分都会从肺菊的方向去寻找。
那他们的话，比如说是你刚才讲的是皮肤，那皮肤上面的话，就他们之间会互相 interfere， 就是你都有 staph 那个是stimulant 是吧？或是 staph aureus， 因为经过这些 antimicrobial 的分泌而造成互相间排斥，是不是有这种想法？
对，的确是 normal flora 之间有一些竞争。那另外一个 idea 是说，肠胃道菌相也是有这种效果，就是好菌坏菌竞争。对
，OK， 不一定是好的坏的吧，就是有这种，
有这种效果。
应该是讲说是 commensal 或者是 commensal bacteria 或者 pathogenic bacteria 之间，也许那个 pathogenic 在某种情况下，它会让 commensal 就是比较不在，就是比较会 dominant， 那个 pathogenic 会 dominant。像 staph aureus 事实上是平常我们皮肤上面没有那么多，但是有些病，比如说 atopic dermatitis 异位性皮肤炎它就会多，那也许它有这方面的，它就会让这个另外比较多的 staph epidermis 让它变得少的，大概有这种现象。那我想再问就是想要做 fusion 的，然后让以后能够当药用是？
我们原本应该说现在的 idea 就是试着让它有2种功能。对，就是所以我们才会选一个针对刚刚是阳性，一个阴性。对，
那这总是希望说它可以拿来 treat。
对，是希望说如果因为其中这个 lysozyme 的部分，根据过去的研究，他们有很多已经有点想要把它转为药用，所以做了非常多的修饰。对，那我们这一次想说，既然可以修饰，那也有前面的前人的研究的话，是不是说，针对不同的细菌，透过一个 fusion 或者怎样就可以，它原本已经可以，也许在血浆里面可以存在一段时间，那我们透过修饰一些别的，让它带有别的功能。因为现在问题就是说，如果短的 peptide 的话，现在应用在血液里面，它还是很容易被分解掉。那透过 fusion 也许可以延长它的half-life。
我想可能还是会有这个issue， 就是是不是能够用在，尤其你如果讲说是要用，就是 infusion 的方法，可能蛮 challenging 的。对，那这里的话，我因为这个领域我一直有兴趣，但是我自己亲自不做这方面的。是的，偶尔会看一些文献，然后但是我就是中研院是有2个团队，应该不止了，但至少有一个是细生所的陈志毅吧，你有没有听过他？他实际上在海洋的那个宜兰，我们有一个海洋研究站，他大部分时间可能在那里，他的 antimicrobial 是从鱼类那边分出来的。对，我想据我知道他也对 对制药有兴趣。那另外是我过去在生医所同事，他已经退休了，叫廖伟弟。他在退休前的大概3-5年，也是往这方面制药方面走。他就是说，他想在这个 catheter 上面 coat 这些 AMP， 然后那就是让那个比如说尿道管还有什么的这个心脏这个，但主要是他所以说尿道这一方面就不会就是不会被 bacteria colonize 这种做法。所以如果有兴趣的话，我觉得这个领域是还有很多可以做啊，而且蛮 challenging 的。所以如果找到一些可以就是 discussion 啊那现在我廖伟弟已经退休，他已经不再做，不晓得他也没有继续在这方面，也许跟业界一起做发展，但是他应该会很乐意分享一些经验。对，我想所以有兴趣的话，我可以做introduction。对，那另外一个我想到的是，当然就是你想要 propose 已经够多了，所以并没有建议一定要往那方面做。但是这些不少的 antimicrobial peptide 对这个 host 的会有影响 ，inflammatory response 啦，对，是不是 inflammation 有一些或是有些 disease，inflammatory disease， 像皮肤我就比较清楚是像rosacea， 后来有个也许你知道这个团队在 UCSD 的，他是皮肤科医师，叫做 Richard Gallo， 他发表不少 antimicrobial peptide 的文章。那后来他在一篇应该是 Nature Medicine， 他发表的是这个对这个 rosacea 它的好像是哪一个 LL37 还是什么，对它造成这个皮肤的 inflammation。被认为那个是因为细菌产生的，不对，它这个是自身的 antimicrobial peptide， 对，应该是 keratinocyte 就会分泌，然后它当然是 antimicrobial， 但它同时也会是对 host 自己本身会有影响，不见得是 antimicrobial。那我现在问的事实上是我的 question 就变成。这些 bacteria 都会，我们比如说 commensal 的话，它有这些 antimicrobial peptide protein， 除了对其他的 bacteria 有抑制的影响之外，它可能对 host 会有一些影响。有这方面的，你刚提的这个 lysostaffing 有这方面的资讯吗？在 host。
你说对 host 本身也许有一些发炎或什么的影响？对。目前比较少看到，大部分都是
bacteria 之间，
就是应该说大部分都是
antimicro。
应该说那个 protein lysostaffing 这个 protein 它本身应该说它目前被使用的被做一些 modify， 就是fusion 或加一些 albumin 之类的，都是可以都直接被用在动物上面，所以比较少提出说它本身会去影响宿主的一些发炎
这边是可以考虑的。当然，我就说是这种的话，我今天提了好几次合作，就是并不是说是这个团队就要做，因为实在是要做的事情很多。但是如果有觉得说是值得的话，因为Staph aureus， 我想看啊，它你这个主要是 Staph stimulant 的分泌嘛出来的 lysostaffing， 然后它会针对 Staph aureus 会 kill。那是我有点 embarrassed， 我不太知道 Staph stimulant 它通常在什么时候会看到它，是不是可能有些皮肤病的时候它会出现？那如果有的话，我就建议说是不是可以考虑它对 host 有没有影响？Staph stimulant 我是这个顺便学一些，就说是在什么样的情况下它不是 commensal 嘛，它不是 commensal bacteria， 它是什么？我真的应该知道，但是我没有这个 information。Staph stimulant， 它是 皮肤上面可以看到它肠道菌，它是
哦，
或者有动物有。没关系，我自己去查就可以。那个我在另外一个在等于说是不是考试了，就是我是怎样了解，我这个也是一面听一面学一些，所以是我对 gram negative 有兴趣的话，像Acinetobacter 是intracellular 的 bacteria 吗？是不是 intracellular？ 像 Listeria 啦、Salmonella 可以进到细胞里面，还是不是？
那个院士，因为不好意思，梁同学因为他这个算是有几个 lab 的一个 collaboration， 所以这个substance 或者是说这个 antimicrobial 的这个 fusion protein 的当初的 extraction 是从我们慈济大学这个medical technology 的那个 lab 所做出来的，所以他现在是比较做后面的 modification， 所以这个比较 origin 的部分他也有点不太清楚，所以我们就可能也让他回去再去消化一下。
我只是这些对我都非常有趣的一些，
是，
这也许能够学到一些，但是我已经学到不少了，所以是没问题。那这个我也可以也去查一下，因为我们现在很希望多知道哪一些 bacteria 是 gram negative 是 intracellular， 对。那刚才讲的这些 antimicrobial 是对 host 的影响，我觉得是很重要。对，
我想刚院士所提到的，其实在一些 in vitro 的 study 里面，目前看起来是确实是有一些这个 antimicrobial 的一些 function， 但是in the future 如果是要做到我们的就是不管是人体或者是环境使用的话，我想那个 toxicity 的 safety 那些可能都还是要 pass 才有办法。
对，那除了那个之外，如果说不往制药那边走的话，就是它本身就是 bacteria 会造成 host， 就是 host 的 microbe interaction。那这个之间就是从 host 是 microbe 的micro host microbe interaction 的情况下，那 microbe 怎么影响到 host？ 它可能可以用 antimicrobial 这些 peptide 或是 protein 影响到 host。对，我并不是说制药的时候要注意，那当然就变成制药的时候也要注意到这一点，就它会实际上是会造成发炎反应，很可能会除了刚刚讲 stability 的 issue， 但是我是觉得说是因为这些如果这个 bacteria 本身就在我们的这个皮肤上面啦、肠道有存在的话，它可能在某种情况下会影响到 host。那这些 antimicrobial peptide 就会是在有这种双重功能，它一个是 kill 它的伙伴嘛，就是它的 microbe， 但是它同时对 host 的影响。对，那个是可能做的不多，但是我刚刚提到 Richard Gallo， 他做了不少这方面的，就是所以它有双重的功能，它对 host 会有影响。对，
我刚刚好像记得院士所提到的是在 UC San Diego，right？
对。
OK， 那我会再请那个梁同学这个部分 maybe 再 trace 一下这个部分。
有一篇文章是在 Nature Medicine 发表。
Yeah。
对，我还写了一个 commentary。
好，所以如果有这个需求或者是一些 information 的话，可能我们再多 search 一下。OK， 好。那今天非常高兴有这样的一个机会，虽然我们是 online， 听了院士您的这个 presentation， 然后也让我们的同学有一些机会可以跟您做一个报告，然后也有一点 brainstorming 或者是给一些in the future 的 research 的 direction 哈。那我想今天我们大家都收获蛮多的。但是也辛苦院士您今天整个下午就陪着我们这样子过了一个下午。好，
没有很高兴有这机会。对， 对，而且这个在线上的话也很 effective，就是就可以跟大家 share 我的 research， 又可以听到大家的 research， 然后一些互动，我觉得这非常好，谢谢你的安排。那问候那个院长。
好，我会跟钟院长讲一下，他虽然今天没有办法到场，不过我们 final 这些的 information 都会传递给他。那也希望下一次有机会的时候，再请院士来到我们花莲走走，好，然后我们再做一些
期待，
现场的communication。
好的，
好，那我们是不是把镜头到我们所有在场的同学跟老师们？那我们是不是最后跟我们院士说一声谢谢，然后也希望有一个have a good weekend 哈，因为今天是 Thanksgiving， 那同样也是也是一个good weekend 哈。那我们谢谢我们的刘院士 ，OK，thanks for the Professor Liu's the comment and today's talk. So today is a Thanksgiving holiday in the U.S. but the same in Tzu Chi, we always say 感恩 right? So Thanksgiving is more like the goodness and thanks everybody. Okay, so thank you, the Professor Liu.
Thank you.
I'll see you next time.
Yeah, see you next time. Yeah, bye. Thank you. So 非常顺利，对，
是，那这几天会开始会变冷，也请院士要自己多保重。
大家保重，对。
好，那我们就下次见喽。
下次见，
谢谢，拜拜。